Kia ora koutou, welcome back everyone. Hope you've had a good lunch. I'm going to promptly hand over to mana rangatira member, Jan Hania, who's going to bless the next session and with karakia and give us a welcome back. So kia ora, Jan.
Inga mane i ngā reo, aku nui ako rahi o te motu nei, tēnā rā tātou katoa, he mihi whānui kia koutou mō tēnā iahiahi, mō tēnei kōrero, whakahirahira, whakaoranga, whakakaiteaki nei o nā tangatuki ho, he karakia timatanga. Inoi tātou, tāne mātua, tāne nui arangi, tāne whakapiripiri, whakarongo mai. Mai wehinga o mātua, nau ki te pō, nau ki te ao. Nau e whakiiri te whānau marama, nau ko te awatia. Nau e tiki te mātauranga, nau ko te wānanga. Nau e pūpuhi te hauora, nau ko te tangata, nau a hau. Ati hei, mauriora ki te whenua, mauriora ki te tangata, mauriora ki a koutou katoa, tēnā rā tātou katoa, kia ora mai tātou.
Ngā mihi, Jan, thank you, kia ora. So before we head into this afternoon's theme, I've just got a few housekeeping notes, and I really should have said this earlier. So we have a lot of fantastic prizes up for grabs, and you become eligible to click on the win points button and get involved. So, you know, you snooze, you lose, click that button and get winning. So kia ora to that. Also, another function that we have here is the meeting hub. So if you want to meet up, get into that hub. So we have the Q&A button. So for the afternoon, if there is some burning issue or question, or specifically to the panel or the speakers or the whole forum, please use that to get in touch. So kia ora to that. So I think I will leave those MC duties alone now, because there's something far more blessed happening. I'm going to introduce our session chair, the amazing Sophie Jerem. Welcome. Sophie works with me in one of the NRT projects, Toi Tau Whakatairinga. So it's a great honour to have you here as our chair today. You're also a community activist, an arts curator, and whose doctoral thesis focuses on the role of art practices in forming shared spaces. So you're a research fellow in the NRT program and implementing a creative capital strategy with Wellington City Council. How does that feel, Sophie? Welcome, no mai haere mai, as our chair this morning.
Kia ora, Nick. Thank you. No, it's great. It's great to be across different worlds, as you know.
Yes, it's a nice journey doing that, isn't it? You don't have to stay on one world too long. You can journey across to another. Some say it's like piwakawaka pecking the forest floor to see what grub is juicier. However,
Yep. Or just getting the Carrera's view, the widest view possible, I think.
Oh, that's a good perspective. Yes. So, welcome as our chair this afternoon, and I'm also going to do the honour of our next speaker and introduce Jack Craw. Now, I think Jack Craw needs no introductions because everyone knows who Jack is, but I will give the formal introduction. No mai haere mai kare. It is a great honour that Jack is here with us today. He was my boss once, and still is really. Jack was a biosecurity manager in the Auckland Regional Council that then morphed into Auckland Council for over a decade, though I'm sure it felt shorter than that because it was such a good time being there, before branching out on his own as a biosecurity and consultant for 8 years, which led him to wonderful places such as Pitcairn Island. enough said. During his time, he's also been an integral part of the Biological Heritage Science Challenge as a knowledge broker. And now Jack is currently a Northern Regional Councillor. So without further ado, welcome, nā mai haere mai, to Councillor Jack Craw.
Kia ora, Nick. Kia ora, Jan. Kia ora, Sophie. And kia ora koutou. I probably don't have to say any more about me. Anyway, I'll just get straight into it. I guess I have always been interested in the interface between science and uptake, and how the community can assist with science uptake, and how the science community can help the wider community. So I think it's timely for us to talk about what collaboration means and how we move collaboration into even a true partnership space. So these are my views, and by the way, I mean no criticism of biological heritage. I think bioheritage sets a commendable example in how to do science with the community in today's terms, and in particular with its focus on tangata whenua engagement and involvement of mātauranga. So anyway, I'll kick straight into this. Before you begin your research, this is pointed to researchers. Wrong way. Understand that it's not your science. It's not the researcher's science. It's everybody's science. It sounds pretty glib, but it's often overlooked. You know, a lot of people, scientists think, well, I've got this thing called science, and I'm going to kind of dump it on people. Well, it's not. It's everybody's science. And before you start, you need to identify who your potential collaborators and recipients are. That also sounds kind of glib, but it's not always done either. You need to figure out, well, who's this going to impact upon, and identify them, and then talk to them. Spend time asking them about their needs, because their needs may not be yours. Their needs may be completely different. Their concerns will certainly always be slightly different to yours. The community and all the different sectors, we could be talking about arable farmers, or we could be talking about a hapu and herakino. It doesn't matter. It could be a school. It doesn't matter who they are. You need to spend some time asking them about their needs and concerns if you want to succeed. There has to be something in it for them. It's like everything's a deal. You have to offer them something over and above an urge to assist research. I mean, pure scientists, they're in love with the science, and aren't we all? We're all in love with the research. The research is a purity and a beauty all of its own, but it's not necessarily true for the wider community. They may not even trust science. So you have to find a bottom line for them. What possibly could be personal outcomes, or what could be a basis for co-investment? Okay. The next big thing is where we start to get a bit more of the nitty-gritty. Involve your collaborators or your partners in the research design, the actual design of the research, because then you'll test the methodologies. You'll be able to say, well, are we going about this the right way, or could we improve upon it? You just don't know. You might actually learn something if you test the methodologies with all the participants. You get to what you will do then, you will get to your mutual bottom lines faster. And so your impediments, a lot of your impediments will be removed if you get involved with allowing your participants to be involved with the design. And you'll start faster. You'll start your research faster. You'll get better buy-in. Of course, you'll get better buy-in, and you'll get better research uptake. Not quite necessarily the same thing, but you'll get better buy-in and uptake. You'll get better participant program monitoring. Now, increasingly today, with most science being applied science, it's going out to communities, and we're really anticipating on sometimes relying upon our partners and our collaborators to do a lot of that monitoring for us. Well, if you don't involve them in design, you're going to find it hard to get them to participate in the monitoring of that. But if you do get them involved, of course, it can save you a lot of money. You'll get better political and funding support. And I can say that's a fact at a Northland Regional Council level. And also when I was Biosecurity Manager in Auckland for 11 years, we're the same kind of we're in the same position, in that if research comes to us involves us, the end user, with the design, then you're going to get much more likely to get funding support and also political support, which is a sort of area I'm in now. And it's not always easy to get political support, but if if you find your whole team's involved with designing the program, then you're much more likely to get political support. You'll get better program results. There's almost no doubt about that. I mean, if you use examples like Kauri Rescue and also our biological heritage, Kauri dieback and myrtle rust programs, they've performed really well in all these spaces, and they're certainly going to get better program results, and they'll get more program longevity. It'll also awaken interest in science amongst iwi hapu, amongst regional councils, are the two sort of obvious areas that I see. But also, I mean, I don't want to speak for DOC or anything like that, because I think they've got a reasonable focus on science anyway, but I'm not sure if regional councils have quite the focus on science that we'd like to see. And of course, it'd be lovely to awaken an interest in science amongst all the many iwi and hapu. I mean, there's 210 hapu in Northland alone, so it is a monumental job, but if you're going to be operating in this space, you need to talk to them. And if you do that, especially if you're talking to iwi and hapu, then scientists are more likely to embrace mātauranga. There's no doubt about that, because you're getting a two-way dialogue and how you design your programs, and you might learn something about how to design a program, or even how to go about thinking about what the needs are. The next thing you need to do, you absolutely need to empower your collaborators. And empowerment doesn't just mean praise them up. It means a whole lot of things. That means your partners, your collaborators. And I think the future, and right now even, includes budgetary and managerial responsibilities if they are sought by those participants. What we're talking about now is co-management of those programs. It may be seen as a step too far for a lot of scientists to have to actually share their budgets and share the management of those programs, but in an ideal world, that's what you will have. So I think scientists now need to be thinking more about how can we involve our partners in actually doing the delivery of the science, even if it's just the monitoring part. It may not be the empirical parts of it, but you may have to consider giving up budget. Okay, that will almost certainly require better audit. And I know a lot of people don't like being audited, and a lot of organisations don't like being audited. But if an audit is part of the ground rules, then it's not a problem at all. Nobody should ever be afraid of audit. It's part of community and personal growth. If your program can withstand audit, then it's a noble. Well, it's roadworthy. So I think we need to look at where you can. Everybody needs to have their cards on the table, but we're already all able and ready to share the responsibilities and the budgets. This also encourages growth in citizen science. Now, we will talk about citizen science. We talk about it as like some wonderful thing, and we're getting quite a bit of it, but I don't think we have as much citizen science as we really need in New Zealand. And a couple of reasons for that is it's cheaper, because we all know that science doesn't get funded anywhere near enough. We're a country that doesn't really value science. That's just an unfortunate thing. We say we're getting better at it, but I'm not really that sure if New Zealand's getting better at encouraging science. So encouraging growth in citizen science is a good two-way thing too, because if you start awakening the value of citizen science and the citizenry, they may start calling for more science budget and more scientists. Also, that whole deal about empowering your collaborators will add to the status of the participants and also their enjoyment. And you should never overlook that. I mean, a lot of citizen scientists or people who get involved with those programs, they see quite a lot of status in that. You know, they feel like being involved as a scientist is quite a noble and a pursuit and something they haven't done before. And it gives them a kind of lifelong, well, certainly a long-term feel-good factor about it. So, you know, it's a positive feedback loop, and I think part of that empowering your collaborators leads to this. And also, just as an overall principle, sharing the credit doesn't diminish the credit. Sharing the love doesn't diminish the love. It only increases it. And it's a pretty mean soul that attempts to take all the credit for a program. They'll probably find that they get quickly forgotten. If you share the credit, you'll find that your own personal status will actually be improved by this. A couple of little notes about other success factors that we get in a shared or co-governance approach to science is, if you want success, find a hero. It may not always be possible, but I always remember we had a scientific feral pig management program at Auckland Regional Council, and we very quickly inherited All Black Glenn Osborne as a pig hunter. Now, he turned out to be a superb teacher, an absolutely unbelievable teacher of the others, and an encourager, and a person who led the others because he had the status. And in the end, we employed him on a UN program. I took him up to Niue to teach them how to do it up there, and he still goes up there and teaches them. And because he was such a hero who was on board, people flocked to him. So finding a hero, it's not always easy, but if you can find a hero, boy, it makes things a lot easier. You have to praise all of the participants. I'm not just talking about some of them. You need to praise them all. It's a responsibility, it's a job, but if you want to succeed, you have to do this. And the reason for that is that when I was working in research in Australia, I did a 3-day course on how to get engagement in ecological and environmental programs, community programs, and then how to retain those people. And we learn every time the same principles. People join a cause because of the cause. They want to save tuatara, or they want to save, a native fern or a habitat, or prevent climate change, whatever it is. They join because of the cause, and that's what dictates their initial involvement. But that doesn't dictate the longevity. People leave because everyone's the same. We all rely on peer acclamation or peer acknowledgement. People leave a program if they feel unappreciated by their peers, and that's a very important thing that a lot of people forget. And I've seen it happen time and time again. I've been involved with things like Forest and Bird and Friends of Matakoa Limestone Island forever, and involving volunteers. And if you keep your volunteers praised, it doesn't mean you praise them to the skies. You just make sure you get the odd photo with everybody in it, and you name everybody in that photograph, and you make sure it makes the local community paper. Just little things like that, because John will be down at the local golf club, and someone says, "Oh, I saw your photo in the paper. Are you part of that, are you?" And I'll go, "Yep." And that's all. That may be all it takes to keep that guy involved with that program for another year or two. So you always have to make sure that people's own personal needs are met by acknowledgement of their work in a program. It's something that's often forgotten, and we just need to remind ourselves to keep doing this. Here's something that gets a little bit harder. You have to make all your publicity a co-publicity. That means like quotes from both sides. If you've got a scientist involved in a fantastic new program, and you've got a hapu, or you've got a forest and bird group, or you've got a community group, or whoever they are, a weed group, make sure that a contact from that other group is in the media guide for reference, so that journos can not only ring the scientist or the person that's funding it, but also the person who represents that other group, so they can have a so you're raising all parties to the same level of status when it comes to being able to talk about the program. And of course, it doesn't mean you get to talk to everyone involved in the program, but there'll be a representative of the people who are doing the mahi. So, and that's one way to keep them involved. And this one here may get a little bit harder. Be prepared to share authorship of reports and publications. I know that Biological Heritage seem to be pretty good at this, but I've seen a few programs where scientists they tend to be a little bit selfish about the authorship of the publications. There again, sharing the credit doesn't diminish the credit. So be prepared to put 10 or 12 names on your publication. There's nothing wrong with that. It can still be Smith et al. And even if you mention 9 other people after that, it's something that they can have for their lives, a lot of people don't ever get their names in a publication, but if they get the names in this one, they'll hold it up and they'll treasure it, and it'll also help their continuing involvement with the program. Just a couple of last points here. If research seeks behaviour change, the first and obvious thing, get a social scientist in your team. I know that's it's become quite a feature and a damn good thing too, because until maybe 6 or 8 years ago, there wasn't much social science involved with other sites, and a lot of programs failed because they didn't get enough support, or they weren't really looking at the right cause of the problem, or reaching the right audience, or engaging with the right people to get involved with it, any number of things. But if you've got a social scientist in your team, you've got a much better chance of getting the behaviour change that you seek. And secondly, consider behavioural changes you may need to make yourself. They may be cultural changes. They may be, you may have to think about sharing the things I've just mentioned before. So if you're talking about behaviour change, consider that you may have to demonstrate or undertake some behaviour change yourself. And as an example, mātauranga isn't just about data or information, it's about values, but it's also about behaviours. So if you're going to be working in a mātauranga space, and my advice to you is get some really good advice from a mātauranga proponent on the kind of behaviours that are expected. That may sound a little bit esoteric, but it's not. It's actually very important. If you want to succeed by anybody's terms, you have to understand the value systems and the behaviours that go with that. And just two final points. One, and shoot me down if you like, talk about holistic research and the risk around it. All research today tends to point towards holistic approaches compared with the old empirical approaches. We're all looking at, what's holistic, what's good for everybody, what's good for the entire ecosystem, rather than just trying to solve an empirical issue. But holistic doesn't just mean New Zealand. Holistic means the planet. And I'm not quite so sure that some scientists in New Zealand understand that. And I just pose a question: have novel approaches that are being suggested now considered the IUCN or the International Convention for Biodiversity Protection and the Cartagena Protocol? Because they are important documents that New Zealand has signed up to, and which mean that no partner, no signatory can undertake activities that might might threaten the biodiversity of another country, like a pest here may be a taonga somewhere else. And even if you think that's a little bit esoteric and you don't think that that's relevant to New Zealand, well, my last point is not considering the international picture will likely affect your ability to implement research. When I was working in Australia, my boss there said that, because New Zealand, that was in 2000, said that New Zealand was looking at GE approaches for possum. He said, we'll oppose that because brush-tailed possums are taonga over here. So it'd be a pity to spend $50 or $60 million on your research and then not being able to actually roll it out because of an international agreement. And my final point, it's not actually related to my subject, but it's something that's really become incredibly apparent to me as necessary. Can we please all work towards creating a national biosecurity surveillance strategy? We're all doing so much surveillance, but it's not really joined up. At the moment, and we talk about building on existing work, like botanical societies to do a good job. Birds New Zealand do a fantastic job of what they do. The pine industry does a great job. There are other groups that do work and perhaps like pasture pathogens and things like that, but none of it's really joined up. We need to provide, we need to find where those gaps are. And this isn't just an MPI job, this is a job for everybody. We need to find those gaps, and then create standard reporting formats for all participants. We're going to make it easy for people to participate here. So, it's a big job, but it's just a personal plea for me right now. I know this work was started by MPI in 2006 because I was on a national working group that did a bit of preliminary work on it, but with a change of government, that work was dumped. And I don't know how much work has been done lately on picking this up. So that's my 10 minutes. Thank you. And I'll just like to close with a photograph of my new grandson doing some field work. Thank you very much.
As I said, a virtual paki for Jack. Thank you for those your kōrero. There's a few wero or challenges in your presentation, as there always is. So we're going to have a Q&A. Post your questions or your responses to what Jack has challenged us to think about. The first thing that I thought when you were talking about recognising other knowledge holders, whether they be with in the mātauranga mana whenua community, or whether it's citizen science of communities or wherever, industry, other places and spaces outside a Crown Research Institute or a university, do you think we are educating our scientists of the future those values of where knowledge is, and particularly the rarua around mātauranga versus Western science, reared its ugly face this year or last year, but also the cultural competencies of our scientists into the future, and also really recognising that knowledge is not just in the traditional science system. So because a lot of academics still think that, we have, of course, some treasured academics and teachers who don't, but are we as a country working that out and having our younger generation of researchers with those skills?
I don't think we are. I've been a judge of science fair. I'm an absolute fan of science fair. I've been a judge with about a 10-year gap in the middle when I was in Australia and then in Auckland. But I was a judge of science fair in the 90s here, and then since I've been since I've been back here in 2008, I've been a science fair judge every year. And I have to say the quality of the science fair work is actually falling. It's not actually getting better. I mean, there's always going to be brilliant young scientists doing great work, but they're not doing work that was actually as challenging or as intelligent, really, as work that I saw in the 90s. It's a little bit disappointing. And there's always great kids that come through and that they're going to be great scientists. But overall, I think the schools aren't focused on science has dropped away. I don't know why that is. I'm not a teacher. But that's the downside. Your question was, are we teaching appreciation of science? I don't think we've kept up in New Zealand. As I say, I think it's become depressing that New Zealand has really become a country that doesn't value science anywhere near enough, and that's reflected in science funding. It's reflected in how we recognise our scientists. We don't really. But I think the good news is, though, Nick, is that I think Mātauranga is making a real bid right now. I see Mātauranga popping up everywhere, and it just gladdens my heart. I see it popping up in schools. I see it popping up from the flax roots. I see it popping up everywhere. And I see it, obviously, it's a big part of biological heritage, but it's also a big part of government funding. So I know Tangata Whenua have done something that us Pākehā scientists haven't been able to do. They've grabbed the government and they've got the government to focus on their worldview and on, okay, a lot of it's about a case of trying to catch up 200 years of colonisation, but I think Maori Tangata Whenua was doing a pretty fair job. You have a look at all the new funding, whether it be and even the aspects in new legislation like the national policy statements on Indigenous biodiversity, the Te Mana o Te Wai programs. They've all got a huge focus on Mātauranga. So, boy, is that heart-gladdening stuff, because Tangata Whenua are going to set the standards for environmental performance for the rest of us. And man, is that something special. But are we doing enough to bring the population across in terms of cultural awareness of the needs? I don't think we are. I think what's going to happen is that it's a bit like talk against colonisation. It's going to result in a little bit of inevitable backlash from those who have never understood or don't want to, and that can happen. That's going to happen to Mātauranga. It's absolutely going to happen. I think we've been lucky to date. I don't think we've had too many of the white bread brigade attacking Mātauranga, but it will come. So I think there's a big need in the university system and also the high school system to talk about the cultural talk about what Mataranga really is, I guess, and we need to get people like Toby Ashby out and into the schools and the universities. I mean, if we could clone them, we would. But I think, yeah, you did right. Anyway, I think I've answered as much as I can.
Yeah,
Thanks.
no, thanks. Yeah, kia ora, Jack. One quick one here, Rebecca McDougall, kia ora. She loves the idea of find a hero. So there's someone, what qualities do you look for in someone when you're looking for an ambassador for your program? Good question, Rebecca.
Yeah, tough one. Obviously, someone who's imbued with it, who's on board, but somebody who's willing to roll their sleeves up, all that. But a hero really has to be someone who's pretty well known and really liked. You know, I always remember back at Auckland Council days, Nick, we used to spend a lot of time trying to find that person that we could get on board to help us with the Kauri program, and we went through all the current pop stars and All Black captains and whatnot to try and get them to embrace the program, and we were semi-successful. I think we probably could have done a lot better, but it's not just political support. If you've got a community leader, a person that is a thought leader in some way, it might be, I don't know, Sam Whitelock or something. It could be Benny. I don't quite know who you best go for. It's just somebody who's, what do they call it? An influencer. That's all they need to be. They don't have to be an intellectual. They just have to be an influencer who says, you know, I think that Myrtle Rust program is fantastic and I support it. Next, and it could be, as I say, it could be Lord, I don't know. They just, I guess, Rebecca, to answer your question simply, they just have to be an influencer, really. They don't have to be deeply involved. It'd be nice if they were, but even if they're just someone who mentions the program as being noble, that's actually 3/4 of the battle won. Thank you.
Kia ora, Jack. One more quick one. Bill Dix put in a few. Thanks, Bill, but a quick one. Where would you put the biosecurity surveillance effort? Would it be look for the current pests we've got versus the new and emerging?
Oh, no, absolutely new and emerging. Thanks, Bill. Great question. Absolutely the new, the unknown. It's all about first records. It's absolutely about, surveillance has got to be about the future. It's got to be future focused. Well primed. Thank you, Bill. I should have mentioned that. So thank you for that little prompt. Yeah, absolutely. It's got to be for the first records, like what the botanical societies do for, like the herbarium records. Of course, first records are incredibly useful to plot trends as well as being useful for their own reasons. So, yep, absolutely future focused, Bill.
Yeah, kia ora, and definitely agreeing with the National Biosecurity Surveillance Response or kaupapa. And we've got some deep dives from Desi, kia ora Desi, around, and Kate around the balance in the Western science system and mātauranga Māori, but I don't think we're going to have the time, and that's a real deep dive. We should have a think tank online or kōrero around that whole kaupapa. So aroha mai, Kate and Desi, but if we went down that, Sophie and our wonderful presenters wouldn't get back on the stage. So anyway, ngā mihi more for whakaaro, Jack. It's been great having your presentation and attending this afternoon's session. So kia ora.
My pleasure. Thank you.
So I'm going to bring back our chair, Sophie, who is going to take on her chairing role into the into the next theme and present our the theme and its speakers. So kia ora, Sophie.
Kia ora, Nick. That's good. Kia ora, Nick, and kia ora, Jack. It was great to hear you. I think your challenges were excellent. And the nature of how we practice research as much as anything. Today's theme, this afternoon's theme, is around risk assessment and ecosystem impacts. And from one of the themes of the bioheritage, it's about understanding the kauri dieback and myrtle rust better in protecting our ngāhere. So the goal of risk assessment and ecosystem impacts team is to identify the effects of the diseases and management and as well as which species and ecosystems are most at risk. By understanding these things, then it, I guess, allows us to better prioritise conservation efforts and management decisions. So it's about gathering data and be better informed for the future or 100 years' time. So the first of our speakers on this topic today is Toby Elliot. But actually first of all, before Toby, we will be playing a video made by the team about risk assessment and ecosystem impact. Thank you.
Kia ora, ko Andrew Barnes tōku ingoa. I'm a senior lecturer in community ecology here at the University of Waikato. Not just interested in how ecosystems kind of work, but also looking at how we can make sure that they're preserved for future generations, like for my daughter. Really, theme 3 is about trying to take this whole ecosystem approach to understanding what the impacts of kauri dieback and also myrtle rust are. My role in the project is really looking at the food webs that are impacted by kauri dieback and trying to pull together data that's been collected from different researchers and look at the food web as a whole, so being able to start answering questions about what happens as a result of kauri dieback infecting trees and particularly the below ground impacts to soil food webs. So all living organisms on this planet rely on energy. It's certainly one common thing that we can say about all living organisms on the planet. They need energy to survive, to grow, to live. And I guess one thing that really draws me to studying food webs is that food webs give us that description of how energy actually flows through the system. So they're describing all of the feeding interactions of organisms in ecosystems. And so using these food webs, we can see how energy sort of enters the system from organisms like plants, and then how it's taken up by these higher trophic levels, so these consumers that feed on the plants, and then the predators that eat those consumers. So I think in a way, at least from my perspective, to really understand an ecosystem, I think at least one of the largest scales, we really do need to be looking at food webs to understand how energy is flowing through the system and to be able to really describe how organisms are interconnected. So what happens if we lose kauri from that system? Well, we know that kauri are connected to all of these other species. They have a really strong role to play in structuring the way that the soil looks, the soil chemistry, the structure of the soil. And so of course they're very strongly interconnected with all of the organisms that live in the soil, that live in this very specific and unique kauri soil. I was particularly drawn to the project as well because if we start to lose kauri from the system, the big question is what's going to happen to the rest of the ecosystem as a whole. And so I think the first place to look in a way is the soil.
Here at Waikato we're looking at the soil invertebrates. But there are other groups within the same theme that are looking at soil microbes and soil enzymes and litter quality. But here we're only focused on the nematodes, which are microfauna. So they're the smallest thing we're looking at. And then the mesofauna, which are up to 2 millimeters, mostly calembla and mites. And then there's some macrofauna, which includes like spiders and centipedes, the larger invertebrates. Soil food webs are not only based on the microbes, they're also based on the litter that they receive from the trees. And so we're interested in knowing whether the soil food web or the community of animals that lives in the soil underneath kauri trees is different from non-kauri trees. And then we're also interested in finding out how that might be affected by kauri dieback. So when a tree is slowly dying, the amount of litter and the quality of the litter falling from the tree might change. And so that might affect that might have a whole cascade of effects on the food web.
It's incredibly fulfilling. It's amazing to be able to get given the chance to go into these massive kauri forests. I've never seen so many kauri trees together before. Someone that's quite new into the science world. It's nice and it's rewarding.
This one smells really nice.
It's incredible. It's a learning experience. Yeah, I get a lot of joy from being in the field. I don't do it for the work. I love being out in the bush and in nature and immersing myself with all of that.
We can think of plants kind of like the gateway of energy into the system. So they're such an important organism because they're able to actually fix energy from the sun, which of course we can't do. So plants are able to fix that energy as sugars and carbon, and they can take that up into forms that other organisms are able to eat. And so they're kind of in a way like the gatekeeper of energy into the system, and that of course makes really key species like kauri extremely important. So they're the ones that are really importing energy into the system, and they're making it available to all these other organisms.
The sample that we have on the screen here is this particular one. And it comes from the infected cascades plot. And so this is collected, we get the 5 centimeter soil core, and then that's extracted in the heat extractor.
One of the biggest challenges of getting these organisms out of the soil to be able to study them in the first place is when you go to the field, you collect this soil core, and it looks like something that you know there's life in there, but you can't really see it. And to open that up and try and pick through that and hand collect all of the organisms from that soil core isn't impossible, but it would take you a lifetime to try and do that with the number of samples that we're processing. And of course, some of these organisms, like nematodes, we can't even see with the naked eye, so you would never be able to manually sort of collect those. And there are different ways to do that, but what we have here are these heat extractors that actually we can process up to 86 samples at a time. So it allows us to really process a lot of samples, so that gives us the ability to study a lot of different systems at the same time and collect a lot of information that gives us much more power in what we can sort of conclude from the data. So they actually heat on the top and they cool on the bottom of these soil samples at the same time, and they're controlled by a temperature control unit. So we just set a program, it sets this temperature ramp so it slowly warms up over the period of 10 days. And as it heats the soil from the top, it begins to dry out, and then the soil organisms have this innate sort of response where they try to go down. So if they're outside in the soil, if there's a drought, organisms will go down to get away from that drying soil. So in the process, then they sort of fall out in the bottom, and we can then collect them in these vials, which we then can put under a microscope and study those organisms. To my knowledge, we're the only laboratory in New Zealand that actually has this full setup.
We clean up the sample under the microscope, and then we collected them all into a smaller vial. And then now they are all together here without any soil around, or with very little soil around.
So we're now working on setting up a monitoring tool that will allow us to actually detect the feeding rates of soil organisms. So without having to constantly be extracting soil organisms out of the soil, say a few times a year, every year, that involves taking out soil constantly from the forest, which we don't necessarily want to do on the really long term. So with this method, it's putting these bait laminar strips into the soil. So they're just little strips of plastic that have like a standardized food that is sort of presented to soil organisms, and what we can do then is monitor feeding rates of these organisms in the soil in the long term, And that's something that communities are really easily able to pick up as well, because it only requires sort of a minimal amount of training. So you don't need to be say a trained scientist to sort of, or have any kind of fancy equipment to be able to do this. You just need these strips. You can put those in the soil and use those to monitor feeding rates of organisms, and we can then start to pick up much more sort of high resolution information over the year. So we can look at seasonal differences, and we can also look at how those might change over time. Because it's quite a cheap and easy method to use, we can hopefully carry that on into the future, and that's something that hopefully communities will be able to pick up as well. Research funding is always limited, and I think always the big challenge as scientists is trying to figure out how we can continue research beyond the life of a funded project. And in many ways, one of the best solutions to that is handing over the science to communities to be able to carry out themselves, and I guess in a way be empowered to be in charge of that data collection and keep learning about the systems themselves. It's hard to know how much information we really need to develop a baseline picture. So I feel like this is really just the beginning, and that, from the data that we've already collected, I think we can already start to say something about, so we've mostly been working in the Waitakere ranges, where we can already start to say something about the structure of the soil food webs and energy fluxes in those systems based on the time of year that we've been in to sample. But of course there's seasonality in these forests as well, and so and also there's going to be changes from year to year, just because of some years there might be a drought, or some years it might be a really wet year. And so I think to get really good baseline data, it does need to be a bit more of a long-term monitoring process. I am interested in all sorts of systems, but I always kind of come back to forest ecology. I guess partly because I spent a lot of time in forests as a child, and growing up, when I was sort of old enough, I started going tramping on my own, and just I always found them kind of maybe a little bit mystical and just an amazing place to explore. And I guess I always sort of felt some sort of connection to them, in that I think I always sort of had this appreciation for the fact that I guess we're part of the system, and so being in forests, I actually always felt very comfortable. Of course, when I go into a forest as a scientist, my brain immediately starts working at kind of looking at those technical details about a forest. So I do look at them differently now, but that sort of feeling of mysticism and that sort of connection to forest hasn't gone away. So there's something about forest. When I go into like an ancient kauri forest, it just feels incredible, and it's just one of the nicest places in the world to be.
Kia ora. Thank you, team, for that beautiful video. Lovely introduction. Now, Toby Elliot is a PhD student at the University of Auckland, looking at the impacts of kauri dieback on the dynamics of populations of kauri, using a variety of modelling techniques to achieve this. So we look forward to hearing from you, Toby.
Awesome. Thank you so much. Kia ora koutou. Thank you for coming virtually to my talk. I'm a current PhD student that is basically focusing on kauri population dynamics and basically assessing whether kauri will survive long-term in forests affected with kauri dieback. So kauri is ecologically and culturally a really important tree species to New Zealand. Unfortunately, 99% of its old growth range has been lost through factors such as logging, habitat clearance, and fire. Luckily, it is now protected, and there's quite a bit of secondary growth of kauri going on. However, now it is impacted by kauri dieback, which is a pathogen that spreads through the soil and causes thinning, bleeding, and death in infected trees. So there is currently no cure and no evidence of complete recovery in infected trees. And most of the research on this plant pathogen relationship has focused on the impacts at the individual level. and there's been little work done at a population level, and answering questions like, will there be local extinction of kauri and so forth? And this represents a major knowledge gap. Researching this will basically help us gauge the severity of the disease and help prioritize management regimes. So with many plants, you can go out into the field and you can measure their whole life cycle over 1 to 2 years, and you can look at these population changes quite quickly. However, it is a bit different with kauri. They have a modal lifespan of around 600 years, and individuals can live well over 2,000 years. So 2,000 years is about 500 times the length of the average PhD project. So it's quite impossible to predict and measure these kauri population changes in real time. So what we need to do is use modeling to predict how these populations are expected to change. And to do this, we need to basically get data from all life stages of kauri. So all this from the small seedlings to the massive trees. So what we can do is we can actually use what we have learned from our COVID response to help us with our kauri dieback response. So for example, a big talking point from last year was the R value for the latest outbreak in August and keeping that below 1. However, we can use this mindset and switch things around a bit to focus on the population dynamics of the host. So we can use what's called Lambda values, which is very similar to an R value. But in this case, we can basically go through, and our goal is to see whether this Lambda value can remain above 1 in these infected populations. And yes, that will be the goal of my study. So this brings me to my thesis, which basically aims to assess the impact of kauri dieback on the population dynamics of kauri. So first, there is little data on kauri population dynamics in normal or healthy settings. So we need to establish that. And then what we need to do is how we are doing this is just basically remeasuring permanent plots in the Waitakere ranges and just assessing the fate of different sized trees over about a 10-year period. Some of these plots are symptomatic and with many trees that are showing some strong symptoms of kauri dieback. So that allows us to compare. So we basically look at the three vital rates, which is fecundity, growth, and mortality, that basically govern population dynamics. And our big aim is to assess how these differ between asymptomatic and symptomatic kauri stands. So I have got a few results from 2 stands already, and I will share them on the next slide. So here's data from 2 slides. So first things first, mortality rate is in 6 years, 13.7% of the trees died, which is quite high and quite alarming how fast that is. And these were trees of all lives from 2.5 centimeters to about 50, 60 centimeters. And it's spread quite equally, whereas in the asymptomatic site, it was small trees and it was less than the percent. Growth rate was higher in the asymptomatic site, and the canopies were also a lot healthier in that site as well. So to finish with, my next steps is just basically to finish collecting data from the rest of the sites and do what's called matrix modeling, which basically allows us to predict future population trends and calculate that lambda value for all our populations and just compare between all these sites and disease statuses. And another big thing is we're going to look at the role of competition in dieback on the regeneration potential of kauri seedlings, especially in infected stands where you would expect them to have a slight advantage from the increased canopy gaps that might be produced by dying adults. And we're just having a look at that role of those 2 factors. And yes, thank you for listening to my talk. And yeah, let me know in the Q&A session if you have any questions.
Thanks so much, Toby. It's great to hear that something's good's come out of COVID in terms of thinking about disease. We've got Tomas Easdale next. Tomas is a forest ecologist at Manaaki, Whenua, Landcare. Research's main interests lie in understanding the structure and function of natural forests and ecosystems, helping to find answers and solutions for their improved management and conservation. He's been assessing and investigating carbon dynamics of natural forests in the shrublands and involved in knowledge transfer exercises with government departments. I'm looking forward to hearing about that in connection to their policy and reporting requirements. Thanks. Tomas.
Kia ora koutou. Thank you. So I'll be sharing some results from recent research. Situation is Myrtaceae are an ecologically diverse family. And in New Zealand, they span from shrubs and small trees to tall and emergent trees, and from early to late successional species. We have 4 botanical tribes that broadly correspond to 3 of those are shown in the slide. As a plant family, the Myrtaceae are widespread and can be locally abundant, with a distinctive feature of a very high wood density. So given this context, we set up to run a carbon stock take for the family in New Zealand forests. To that purpose, we relied on the Lucas grid of forest sample plots. These are regularly located across the landscape and provide a robust sample of natural forests. In each plot, all trees are identified and measured, which allows us to estimate their carbon mass by means of allometric models. Given that trees are tagged and these plots have been measured twice, we can have a more dynamic assessment of changes over time. Given that the extent of natural forest is estimated at 7.8 million hectares across the country, and given we have this representative sample of natural forest, we can scale up the plot level values and obtain carbon stocks for all natural forests. Some results. As a plant family, Myrtaceae account for 139 megatons of carbon, very big number. That's 9.7% of total stocks in natural forests, and Myrtaceae are the 4th most dominant of a total 55 woody plant families. In terms of tribe totals, the Metrosidae and Leptospermae account for approximately 71 and 29% of the family stock. And with regards to species, Southern Rata and Northern Rata make up the major stocks for the Metrosidae, but Kanuka and Manuka are also pretty high in terms of stocks nationally. Syzygium, which has quite a limited distribution at the moment, has the lowest share of the lot. In terms of carbon flux results, we have a summary shown here where the first panel shows productivity values that result from tree growth and recruitment. The second panel presents tree losses to mortality, and the third panel is a net outcome of productivity and losses. So in that third panel, any positive values would equate to net carbon sequestration of atmospheric carbon into live wood. Any negative values would equate to carbon losses into dead wood. Broadly, what we can see from the figures is that productivity gains in the first panel tend to be compensated by losses. in the second panel with the result that as an outcome, this near zero or a quite tempered carbon change in all groups in the third panel. So of the three tribes, Leptospermae had the highest productivity, but those tend to be, again, outbalanced by mortality with just only a marginally positive net sequestration for the tribe. In the case of Metrocedere, there's a net loss of light carbon into deadwood. That's because of, well, mortality not being quite compensated by productivity. And finally, well, the tribe were quite stationary with low rates of productivity or loss. Another pattern you may see from the figure is that if we compare the Myrtaceae to all other plant families lumped together in the first panel, we see that Myrtaceae tend to have a lower productivity than all other taxa. Right, so there were some surprises from these results. Well, to see Myrtaceae carbon is very significant, but what was notable is that they account for a comparable carbon stocks than the New Zealand polar carps, which are, well, very conspicuous and characterized by large canopy trees, so a lot of biomass in there. In terms of productivity, Metroceders had a notable low productivity, and that may be partly attributed to the high wood densities typical of the family. This heavy wood is more expensive to produce and maintain for the trees. The net moderate net sequestration for the Canucas and Manukas was again surprising and contrasted with our assumptions for this group. Possible reason is for that pattern is that we were looking at established forests, and we know these species are quite successional. So they could be losing as much as they gain as part of a transitional change along succession. So, well, finally, at present, mortality due to myrtle rust tends to be rare and seems confined to a couple of species in the Myrtae. But I guess what we take from these results is any wide-scale mortality in the long term could significantly compromise carbon stocks in New Zealand forests. That would be about it, really. But thank you.
Thank you, Tomas. That was really interesting. Bringing these issues together, which of course carbon is a major concern as we battle the diseases. So thank you. That was very good, and I'd like to see more of that actually. But anyway, back to we have today, as well as Tomas and Toby, we have the team that were presenting in the video earlier: Andrew Barnes, Manaaki Struik, Simon Whenua, and Luitgard Schwingenmann. And they have joined us for a Q&A. We are running slightly over time, but we're going to extend the Q&A for 5 minutes. Question actually I had for Toby, just a brief one. When you were talking about competition, you were looking at, are you also looking at companion species in your sampling?
No, more focusing on basically how this sort of competition is in basin spatial position. So basically, if there's a tree that's surrounded close by neighbors, is that going to grow slower or has a higher chance of dying than trees that have a bit more leeway? So that's what we're sort of looking into, that spatial sort of area.
Fair enough. Great. Thank you. There's a question in the chat from, I can't actually see, it's from Monique Hall. Thanks, kia ora, Monique. And it's to the Andrew's team. In the video, you mentioned the need and importance of long-term monitoring of the soil food webs. How many repetitions of samples are you and the team able to take for this research? You can decide amongst yourselves who can answer that one.
Kia ora, Monique. I'm happy to take that if you like. Yeah, thanks. Great question. I think there's probably a bit of a discrepancy of how many samples we'd like to take and how many we can. But it really comes down to, I guess, like I was saying in the film, a matter of, I guess, the ability for communities to take up that monitoring, because of course, the project does have a limited lifespan in terms of funding, but what we're really hoping to do is, being able to set up enough of a program and a good understanding of how these monitoring techniques work now while we do have the funding running, so that way when we're sort of ready to, I guess, hand that over in a way that it is really usable for communities with minimal amount of training, and hopefully also being able to hand over the techniques in terms of just making the materials for that monitoring as well, because of course that can be quite time consuming. So I think generally speaking, within a year it would be nice to have at least one sampling replicate, I guess, per season to get some indication of what these systems look like across seasons. And as far as how many years we need to be monitoring, I think that's the big question. And ironically, it's hard to know until we start monitoring and look back at the data and see what the trends look like.
Thank you. Anyone want to add to that?
Yeah, I'll just jump in a little bit. One of the things we've really tried to focus on in this research theme is setting up the current state so that we can come back and researchers in 10, 20, 30 years can look back at the data we've collected and be able to compare against the future. So that's really been our priority, is just to capture as much as we can and set that baseline. And the reality is, as Toby mentioned, that these are trees that live for thousands of years. So we may not be able to really see some of the impacts fully expressing for quite a long time, as and we may even see some positive changes in the short term, and then as leaf litter falls, and then and then the canopies, once it's fully gone, then it starts to deplete. So we just don't know. It'll take a long time.
Thank you. A question for Tomas. In your work where you're working across government departments, are you sharing this knowledge about Mutesi with those in government departments who are looking at carbon sequestration?
Well, we haven't yet been sharing this work on the Mutesi study. We've been involved with other assessments with government departments, but I expect, yes, this work in progress, and we expect to produce a publication and make it readily available for all interested parties. Yeah.
Great. Thank you. Final question from the floor, from Stuart Fraser to Toby. Sorry if he missed it, but you described sites as asymptomatic. To a pathologist, this would imply the pathogen is present, but there are no symptoms. Is this the case, or should these sites instead be described as, I can't wind down here, pathogen-free, there we are, or disease-free if the pathogen status is unknown?
I think we've sort of labelled it as this asymptomatic, symptomatic, just to be sort of on the safe side, because we do not know for certain that the disease is absent in the asymptomatic sites.
Okay, good. There we are. Hope Stuart's satisfied with the answer. Look, it's really tricky. I thought we could have a lovely long discussion, but clearly time has kind of gotten in the way. And I'm looking forward to understanding your papers and meet you all in person one day. Thanks to everyone for speaking and we'll see you again. I'll pass over to Nick.
Yeah, kia ora. Thanks, Sophie, and thanks to the Risk Assessment and Ecosystem Impacts team. That's been some great whakaaro and some presentations there. We're going to be hearing more in our last session on integrated surveillance. So have a quick break. Make sure you go back into the program and join us there. And reminding those fantastic prizes, click on the win points button to win. So anyway, kia ora, and we'll see you soon.